So hey students, welcome back to Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. So good afternoon everyone, or rather good evening everyone. So quickly let me know in the chat if my voice check is audible to everyone and presentation behind me is visible to each and every one of you. Quickly let me know in the chat. Am I audible and visible to each and every one of you? And if I'm audible and visible and you're excited to learn something new, quickly green hearts in the chat and we will start today's class, that is Basic Botany Part 2. Let me know in the chat if I'm audible and visible. Yes? One second students. Yes, I am audible and visible. Yes. So in the previous class, we did about your class 11th portion. Yes, we did a bit of your class 11th portion. Now this is your, since this is going to be a part 2 of your basic botany, I will be focusing on your class 12th portion. Yes, some of the concepts of your class 12th portions and some of the concepts which you might not be aware of. I'll give you a quick example, all of you. Tell me, what is the difference between a carpel and a pistil? Anyone can tell me in the chat? What is the difference between a carpel and a pistil? 90% of the students, or rather 50 to 60% of the students, believe that carpel and pistil are actually the same to same, but they are not. So all of these basic terminologies and basic terms we'll be talking in today's class, right? So quickly tell me in the chat, which is the reproductive wall of a plant? Which is the reproductive wall of a plant? If I ask a class 10 student also, a class 9 student also, a girl or a boy would tell me, sir, the reproductive part of a plant is called as a flower. Yes, it is the flower, right? It is the flower. Now, if we divide the entire flower, into two parts, we can divide the entire flower into two parts. First part is called as your non-essential whorls. Non-essential whorls. Now, what are these non-essential whorls? Non-essential whorls are the whorls which are technically not required for reproduction. They might not be required for direct involvement for reproduction. Direct involvement. They will have certain indirect aspects. For examples, petals will help in attracting the pollinators. But are petals involved directly in your reproduction? No. So we have something called as your non-essential whorls. Under non-essential whorls, it can be further divided into two categories, right? The non-essential whorls can be further divided into two categories. That is your calyx as well as corolla. Calyx as well as your corolla. Now, what is this calyx? Can anyone tell me? Any student can tell me what is this calyx? Calyx is nothing but your group of sepals. Group of sepals. Now, what about corolla? Corolla is a group of petals. Corolla is a group of petals. Now, the other part is called as your essential whorls. It is called as what? Essential whorls. Essential whorls. Now, what are these essential whorls? Again, we can divide them into two categories. Under your essential whorls, the first category is your androecium. It is going to be your androecium. It's going to be androecium. Yes, androecium. Now, what is androecium? You might tell, sir, androecium is stamen, right? Is it actually stamen? No. What is androecium? Androecium is a group of stamen. It is going to be group of stamen. Very basics. All these are very basics which every student needs to know who is trying to learn botany. Now, other part is going to be androecium is your male reproductive part. Androecium or the stamen is going to be your male reproductive part, and the female reproductive part is going to be your gynoecium. It's going to be gynoecium. And gynoecium is what? A group of, gynoecium is a group of your carpels. Is a group of carpels. Now, so if you talk about the reproductive part of a plant, it's called as a flower. We have your essential whorls and non-essential whorls. Essential whorls are indirectly involved in reproduction. They're indirectly involved. That is going to be your calyx and corolla. Calyx is called as group of sepals, corolla is called as group of petals. Then we have your androecium, that is group of stamen, and gynoecium as a group of carpels. Now this is your NCERT diagram. All of you know this diagram, right? That is your entire flower structure. Now if you look at the structure of stamen now, if you look at specifically the group of stamen, if you look at the structure of stamen, it can be further divided into your anther. It can be further divided into anther as well as your filament. Anther as well as filament. Let me write that here. Let me write it here, students. Let me write here. Let's take purple. Now, your entire chapter of your sexual reproduction in flowering plants is divided into how many parts? 3 total parts. 3 total parts. The first part is called as your pre-reproductive phase. It's called as what? Pre-reproductive or pre-fertilization. Also called as what? Pre-fertilization. One thing, students. Pre-fertilization events. Now, under pre-fertilization events, we learn the structure of stamen. We learn the structure of stamen. Now, if a typical stamen, students, a typical stamen looks something like this. A typical stamen looks something like this. And if you look at your typical stamen, it is divided into 2 categories. First category is called as your anther, and other category is called as your filament. Second part, anther and filament, anther as well as filament. Now, if you open your NCERT, and look at the structure of anther, you might get a little confused. The typical structure of anther is said to have 2 lobes. For example, this is one lobe and this is the other lobe. So this is the structure, typical structure of an anther. It is made up of 2 different lobes, and that is why. See, when we're drawing it also, can I see? I have drawn 2 lobes here. A typical structure of your anther is made up of 2 lobes, and it is called as bilobed structure. So one lobe is here, other lobe is here. 2 lobes are attached to each other. It's called as a bilobed structure. Bilobed. It is a bilobed structure. Now, if you read your NCERT, it says something very peculiar after this. There is something called as theca. What is this theca? Now, theca is nothing but a cavity. Theca is nothing but a cavity. Now, imagine this is your bilobed anther. Now, inside one anther, there are 2 thecas. There are 2 thecas, which means there are 2 cavities. I'll give you a simple example. Think of these 2 lobes as 2 separate rooms. So this is one entire house. One house has how many rooms? It has 2 rooms. Now inside each room there are 2 windows. There are 2 windows. Window number 1, window number 2. So similarly, inside a bilobed anther, there are 2 cavities called as theca. There are 2 cavities called as theca. So there are 2 thecas inside each lobe. 2 theca. And that is the reason it is called as dithecous also. It is also called as what? Dithecous. Now tell me in the chat, if one lobe has 2 thecas, if one lobe itself has 2 thecas, how many thecas are there in entire anther? Can anyone tell me? If one lobe has 2 thecas, it means there are in total of 4 thecas. In total of 4 theca. Clear? In total of 4 theca. Now inside each theca, you find something called as microsporangia. Inside each theca, we find sporangia. Now, if there are 4 thecas, how many sporangia will be there? In total of 4 of them will be there. 4 sporangia will be there. Clear? 4 sporangia will be there. I hope this is basics is clear to all of you. Now, if you look at your NCERT, after this it talks about your structure of your young anther, that we will study about in your detailed class. Structure of young anther. There epidermis, middle layer, tapetum, all those structures we'll study later on. But right now, this is just the basics. So let's focus on just the basics for now. So there is an anther. Anther is bilobed. It has 4 theca, 2 theca in each lobe. In each lobe. In each lobe, and that is why it's called as what? Dithecous. And inside each theca there is something called as microsporangia. Clear? Microsporangia. Now, can anyone tell me the next concept which we learn in your sexual reproduction of flowering plants chapter? The next main concept which is going to be there is microsporogenesis. It is going to be microsporogenesis. Microsporogenesis. Yes, microsporogenesis. Now, can anyone tell me what does this microsporogenesis leads to? See, the end product of your microsporogenesis is going to be your pollen grains. But before the pollen grains, the initial product of your microsporogenesis is nothing but your microspores. It is microspores. Now, these microspores later form the microspore tetrad, which eventually becomes your pollen grains, Now, these pollen grains in the examination, they can directly ask you, sir, what is the other name of pollen grains? Or they might ask you a question and they might not mention the pollen grains. So pollen grains has another name. The other name of pollen grain is called as your male gametophyte. It's also called as what? Male gametophyte. Also called as male gametophyte. Clear? So all of you will remember that? Right? It's called as male gametophyte. Now, let's understand the next terminology, that is going to be your, obviously, the pollen grains. Let's focus on pollen grains right now. Let's have a deep focus on pollen grains. Can we draw the structure of pollen grain now? Learn now. If you look at the structure of pollen grain, if you look at the typical structure of a pollen grain, it is divided into two parts. Or rather say two separate layers on the outside. Your typical pollen grain has two layers. The outer layer has little bit of ornamentation. It is little stylish. The outer layer is called as your exine. It's called as what? Exine. Now this exine has certain type of chemical. This exine has a certain type of chemical which will help the pollen grain for protection. This chemical is so strong that even it won't be damaged with the help of organic acids or any kind of natural calamity. It is so strong. Now, what is this natural chemical here? The chemical here is sporopollenin. It is sporopollenin. Then we have inner layer. The inner layer is called as intine. Now, in the examination, they can ask you, what is intine made up of? Now, if you read your NCERT, you'll be able to tell the intine is made up of your cellulose and pectin. Cellulose as well as your pectin. Now, the outer covering is made up of your exine, right? Again, you can divide the exine into sexine and exine, but that is not in your syllabus. So, if you look at your outer covering, do you think the sporopollenin will be there throughout the exine? Do you think? Obviously not. If the sporopollenin was there throughout the exine, how will the cell which is present on the inside will even come out? So if you look at closely at the structure of your entire pollen grain, there are certain minute gaps. There are certain minute gaps in the exine of your pollen grain. Exine of the pollen grain. Now those gaps are called as germ pores. They're called as what? Germ pores. They're called as germ pores. Clear all of you? It's called as germ pores. I believe this is also very easy. This is also very easy now. So that is the structure of your pollen grain. That is outside now. But if you focus on the inside, if you focus on the inside of the pollen grain, you will end up seeing not one, in fact, two different cells. You will find two different cells. Now, one is a larger cell. One is a relatively a large cell called as your vegetative cell. Now, this vegetative cell is very special. This vegetative cell is basically like your nutrition holder. It has a lot of nutrition and it will hold up the entire nutrition content. It is called as your vegetative cell. Vegetative cell. Now, if you look at the other part, there is one more smaller cell. There is one more smaller cell which floats in the cytoplasm. One more smaller cell which floats in the cytoplasm of the vegetative cell, and we call it generative cell. We call it generative cell. Generative cell. Generative cell. Now, in 60% of the angiosperms, in the 60% of the angiosperms, we typically find this type of pollen grain. 60% of angiosperms. But in few angiosperms, what happens is this particular generative cell may end up mitotically divide. Yes, it might divide. So in such cases, this is how pollen grain looks like. In such cases where the generative cell divides into two male gametes, in such condition we have in total of three celled condition. It is three celled condition. Clear? That is called as three celled condition. That happens in only in a few cases of your angiosperms. Clear? Now, till here I believe it's very basics, very basics, and you were able to answer most of the questions, right? I can see Meghana answering, I can see Dhanlakshmi answering, I can see Orochimaru. Orochimaru, instead of staying in hidden village, is coming here. Orochimaru is able to answer. So, right? So all of these are very basics. Now let's look at the one more basic structure. Can anyone tell me what is this structure called as? What is this structure? This structure is a typical embryo sac. It is a structure of typical embryo sac. Now embryo sac also has another name. A typical embryo sac has another name. It's called as your female gametophyte. This is your female gametophyte. Female gametophyte. Clear? It's called as female gametophyte. Now students, before we go to the female gametophyte, can we have a small touch or small, let's cover up megasporogenesis? Obviously, if I sit and cover the entire megasporogenesis, it's going to take a lot of time, right? So that let's keep megasporogenesis in detail lecture later on. But can anyone tell me, there is one more term in your NCERT that is going to be megasporangium. Small terminologies we'll study today. Megasporangium. Now, what is this megasporangium? Is there another name to megasporangium? Yes. The megasporangium, which is given you in NCERT, is nothing but your ovule. It is the ovule. Right? It is the ovule. So another fancy name for an ovule. So it's going to be very easy to understand botany if you know the certain terminologies. If you skip certain terminologies, it can become a nightmare later on. Okay? Clear all of you? Now, if till here, if everything is clear, quickly smash that like button. Quickly smash that like button, everyone, and mark your attendance. Now, students, let's understand the structure of your typical embryo sac. If you look at the structure of your typical embryo sac, it is made up of how many cells? Let's count. 1, 2, 3, 4, 5, 6, 7, 8. 1, 2, 3, 4, 5, 6, 7 cells. Sorry. This is going to be one entire cell. So in total of it is made up of your 7 cell. It is made up of your 7 cell. It is 7 celled. But if you look at the nuclei, before we go to the nuclei, towards the chalazal end we find 3 antipodals. We find 3 antipodals. And in the center we find something called as your polar nuclei. Now these two polar nuclei will end up fusing together and that will produce your central cell. That will produce the central cell. Towards the micropolar end, we see the 2 synergids and 1 egg cell. Now at the end of synergid we see something called as filiform apparatus, which helps to guide the pollen tube into the synergid. Clear? So how many nuclei are there? 1, 3, 6, 7, 8. It is 7 celled and 8 nucleated. 7 celled as well as 8 nucleated. Clear, all of you? Clear? 7 celled and 8 nucleated. Now I'll ask you a question which I asked long before. Pistil versus carpel. Pistil versus carpel. What is pistil and what is carpel? Now students, remember, pistil is nothing but your female part of flower. It's the female part of the flower. Then what is carpel? Your carpel is nothing but it is a single unit of pistil. It is a single unit of a pistil. Now let me explain to you in a much more easier fashion. See, now this is your hand. Right? All of you open your hands. All of you keep your hands front. One second, I'm not able to see the chat, students. 1 second. No. All of you keep your hands front. Now if you imagine this is your entire hand. Your entire hand is gonna be pistil. Your entire hand is pistil. Now if your hand has how many fingers? It has 5 fingers. It has 5 fingers. Now think of this, your entire hand, as your pistil. Now pistil has something called as 1, 2, 3, 4, 5. 5 carpels. 5 carpels. So your entire hand is nothing but your pistil, and your fingers will become carpel. Your fingers will become carpel. Now imagine in this pistil, in this entire pistil, there is only 1 finger. Imagine this entire hand, if there is only 1 finger. In terms of this, imagine this entire pistil, there is only 1 carpel. Then it is called as monocarpellary. It is called as what? Monocarpellary. Now imagine in this pistil we have many carpels. Then it is called as multicarpellary. It is called as what? Multicarpellary. Now in this pistil, if all the carpels are completely fused, if all the carpels are completely fused, then it is called as what? Syncarpus. It is called as syncarpus. So in a pistil, all the carpels are completely fused. It is called as syncarpus. Now imagine in this particular pistil, if all the carpels are free, then it is called as apocarpus. So many words, right? So many terminologies. Yes. So that's what it is. This is going to be an entire pistil, and pistil is made up of many carpels. So it is the female part of the flower. It is the single unit of a pistil. So carpel is a single unit of an entire pistil. Clear, all of you? Clear? Did you understand? Clear, right? That is the basics which you need to know. That is the basics, right? Basics. Now let's understand something called as pollination. Very easy. Nothing too much to teach here. Very easy right now. Pollination. Can anyone remind me what is pollination? Pollination is nothing but transfer of pollen grains. Pollination is nothing but transfer of pollen grains from the anther to the stigma of a flower. That is pollination. Transfer of pollen grains from the, this is your anther, this is going to be your, so transfer of pollen grains. Transfer of pollen grains from the anther to the stigma is called as pollination. It is called as pollination. Now students, let me write down few extra points for you. Now under pollination, we have something called as autogamy. We have something called as autogamy. We have something called as getinogamy or neatinogamy. What is, we have something called as getinogamy. Getinogamy. We also have something called as xenogamy. Something called as xenogamy. Now, what is this autogamy? Autogamy is nothing but your self-pollination. It is self-pollination. Self-pollination. That is transfer of pollen grains. Transfer of pollen grains from the anther to the stigma of the same flower. That is going to be your autogamy. Same flower. Now, it is again divided to, we have something called as chasmogamous flowers and cleistogamous flowers. Chasmogamous flowers are basically normal flowers, and cleistogamous flowers are flowers which do not open. Flowers which do not open, and with the flowers which are typically underground sometimes. Underground. So there is no chance of cross pollination in the case of cleistogamous flower. In the case of cleistogamous flower. Cleistogamous flower. We can call cleistogamous flower also called as your closed flowers. Closed flowers. Closed flowers. Clear? It's called as closed flowers. I believe this is easy to understand. Now, what is this getinogamy or getinogamy? Getinogamy is nothing but also a type of somewhat cross pollination. It is transfer of pollen grains from anther to the stigma of a different flower but on the same plant. I repeat again, it is a transfer of pollen grains from the anther to the stigma to a different flower but of the same plant. Remember, of the same plant. That is called as getinogamy. Clear? Now, what is xenogamy? Xenogamy is nothing but transfer of pollen grains from the anther to the stigma of a flower of a different plant. Of a different plant altogether. Of a different plant. That is going to be your xenogamy. That's going to be your xenogamy. It's typically your cross pollination. It's typical cross pollination. Typical cross pollination. Typical cross pollination. Clear, all of you? Now, the next concept is going to be agents of pollination. Agents of pollination. Agents of pollination. I will not write all of them. So, let's just discuss one by one. In agents of pollination, we have anemophily. Anemophily is transfer of pollen grains with the help of wind. Anemophily is transfer of pollen grains with the help of wind. Single L, right? Anemophily. So, it's transfer of pollen grains with the help of wind. Then we have something called as hydrophily. We have something called as hydrophily. Hydrophily is a transfer of pollen grains with the help of water. Then we have something called as entomophily. Entomophily. Entomophily is with the help of insects. Transfer of pollen grains with the help of insects. Then we have something called as ornithophily. 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 That is transfer of pollen grain with the help of birds. With the help of birds. So let me know in the chat, what is the transfer of pollen grains with the help of bats? Let me know in the comment section in today's video, what does transfer of pollen grains with the help of bats called as? With the help of bats called as. Very easy. If you have studied your extra little here and there, you might know this. Okay, with the help of bats. Right. Now, there are certain other concepts which you need to cover in this chapter, that is called something called as artificial fertilization. Artificial fertilization. Now, what is this artificial fertilization? Artificial fertilization, or it's not called fertilization, let's call it hybridization. Let's call it hybridization. Now, what is this artificial hybridization, students? In artificial hybridization, we are adding the pollen grains onto the stigma. But what do we do right now? Before we adding the pollen grains onto the stigma, there are two options. Either you take unicellular or, sorry, no, I'm taking unicellular. Either you take unisexual flowers, or you can take bisexual flowers. If you take unisexual flowers, life is easy. But if you take a bisexual flower, you have to do a process called as emasculation. In the process called as emasculation, you need to remove the anthers. So the process of removal of anthers is called as emasculation. Now, once emasculation is done, you need to completely cover the flower, called as bagging. When you do the bagging, it makes sure that no extra pollen grain is entering inside. Now, once you do the bagging, there is also something called as tagging. You also need to do tags to note down the date, time, flower, which pollen grain did you add. So all that details will be written on the tag. That is called as tagging. That is called as what? Tagging. Now, there is something called as outbreeding devices. Outbreeding techniques or devices. Now, what is outbreeding devices? Outbreeding devices is very simple. Plants are avoiding self-pollination. Plants are avoiding self-pollination. There are many techniques here, so which we will learn all the different techniques to avoid the self-pollination in the detailed class. In the detailed class. Then there is fertilization. Then there is fertilization. Fertilization in the case of plants is called as what? In the case of plants, it's called as double fertilization. Double fertilization. Because there are two types of fertilization which is happening here. One is called as your triple fusion. Triple fusion. Other one is called as syngamy. Other one is called as what? Syngamy. The two type of fusion which is happening. In triple fusion, that is fusion of male gamete with the two polar nuclei. And in the case of syngamy, it is a fusion between your male gamete as well as your egg. As well as your egg. Your syngamy results in the formation of your zygote and embryo, and triple fusion leads to the formation of endosperm. Leads to the formation of endosperm. Students, there it is. I think we have covered most of the basics from this chapter. The other basics, if I have to cover, it's going to be the seed, fruit formation. In seed formation, the seed is the ovules. Ovules, seed, ovary will become fruit. Ovary will become fruit, ovules will become seed. Right? Apart from that, there is something called as your apomixis. Apomixis is nothing but formation of seeds without fertilization. There is also something called as parthenogenesis. Parthenogenesis is nothing but formation of fruit without fertilization. Clear? Then there is something called as ex-albuminous seeds, which is wherever seeds which do not have any type of residual endosperm. Ex-albuminous. X, the person who left your life. Right? X who left your life. Similarly, in the ex-albuminous seed, there is nothing left in the seed. All the endosperm is eaten out. All the endosperm is eaten out. I mean consumed. Now, the next chapter which I'll teach you basics today is Mendel's chapter. That is going to be your principles of inheritance. Now, this chapter has a lot of basics, and we will learn one by one some few basics here and there, which can help you to understand the entire chapter later on. Okay. The first thing is, what is the chapter name? The chapter name is Principles of Inheritance. Chapter name is Principles of Inheritance. Now, to understand this Principles of Inheritance chapter, you need to know which category of science does this chapter come around. The category of science or the branch of science which deals with such information is called as genetics. It is falls under genetics. So genetics is the branch of biology or science which deals with the entire this chapter. The chapter name is what? Principles of Inheritance. And Principles of Inheritance. Now, what is meaning of inheritance? Can anyone tell me what is inheritance? You inherit something. Inheritance is nothing but transfer of certain characters. Transfer of certain characters from the parents to the progeny. I'll give you a simple example. That is, a particular cat will give birth to a kitten. A dog will give birth to a puppy. Similarly, a human will give birth to another human. That is called as like begets like. That is, transfer of characters from a parent to the offspring. That is called as inheritance. But students, humans, cats and dogs, are they sexually reproducing? Yes, they are sexually reproducing. So students, listen to me very carefully now. In the organisms which undergo sexual reproduction, there is sometimes or most of the times some changes will happen. Exact copy cannot be formed. There are small changes. These changes is called as variations. They are called as what? Variations. So in sexually reproducing organisms, there is always variations happening. Variations are happening. Clear, all of you? Now in this chapter, the first thing which you need to understand is what is a character? What is a character and what is a trait? What is a character? What is a trait? Character is nothing but it is a feature of an individual. It is a feature of a individual. When I say feature, I mean a phenotypic character. Let's not tell phenotypic, it can become a little complicated. It's a type of feature of an individual. For example, if I see someone, I'm like, okay, fine, that person is very tall. That tallness is which part of it? When you look at someone, you observe their height. So height can be written here. Now, if you look at a flower, if you look at a flower, the first thing you notice is what about a flower? Which color it is. So color can also be written here. That is going to be a character. That is height or flower color. I'll write flower here. Flower color. Now similarly, what is a trait then? Trait is an inherited character. It is an inherited character. Let me give an example. Here we spoke about height and color of a flower. Here in the trait we'll talk about if the plant is going to be tall or it's going to be dwarf. In the character we spoke about color of a flower, but in the trait we'll talk about which color is it. Is it red color? Is it white color? Or is it going to be your violet color? That falls under your trait. So I believe all of you know the difference between a character as well as a trait. Yes? Now, the next thing I want everyone to understand is what is a gene? What is a gene? There have been many definitions, but let me tell you what exactly is the definition of a gene. Now, if this is a chromosome, okay, let's know chromosome. Let's say within the chromosome, obviously you have your DNA. Obviously, we have the DNA, right? This is your DNA. Now, gene is nothing but a small segment of DNA. If you take a small segment of DNA, that is nothing but called as a gene. A small segment of DNA is called as a gene. Small segment of called as a gene. It's a part of a DNA. Okay, Ravindrani? Yes. It's segment of a DNA is called as your gene. Now, when we speak about gene, how can we not mention about allele? How can we not mention about allele, right? So, what is an allele? What is an allele? Allele is nothing but an alternative form of a gene. Let me give an example. We have one human here and one human here. The two humans are something called as twins. They are called as twins. Now, do the twins look like each other? If they are identical twins, they look like each other, right? So, do they look like each other? Yes. So, your gene also has a twin. The twin of a gene is called as an allele. So, we can write allele as an alternative form of a gene. Alternative form of a gene. Alternative form of a gene is called as an allele. So, every single time when we talk about allele, so we talk about gene, we should remember that it can have two alleles. But sometimes there can be more alleles. That we will learn in your ABO blood grouping. Okay, multiple allelism. We call it multiple allelism. That you learn in your ABO blood grouping. Multiple allelism. But right now remember, allele is nothing but an alternative form of a gene. Alternative form of a gene is called as an allele. Clear all of you? Easy. The next concept which I want everyone to understand is something called as, what is a phenotype? What is a phenotype and what is a genotype? What is a phenotype and what is a genotype? So, phenotype is nothing but your observable external morphology of an individual. How an individual looks from the outside, that is called as your phenotype. What is genotype? Genotype is nothing but genetic makeup of an individual. Genetic makeup of an individual is called as your genotype. It is called as genotype. Then you can ask me, sir, what is genome? Genome is nothing but the entire DNA of a particular human or particular organism is called as genome. The next thing which I want everyone to understand in the basis of this genetics is nothing but what is the meaning of homozygous. Now, what is this homozygous condition? In homozygous condition, the two alleles are similar. The two alleles are similar in shape, they are similar in structure, or they are very same to same. They are called as homozygous in nature. They are called as homozygous nature. Now, what is this heterozygous condition, students? Heterozygous condition. In heterozygous condition, the two alleles of a gene, are dissimilar. They are dissimilar. Okay, they are dissimilar in nature. They are dissimilar. They do not look like each other. They are called as heterozygous condition. I'll give a quick example. Now imagine when we speak about allele, it can be capital R or small r or capital R small r, right? So when we have in homozygous condition, In the case of your homozygous condition, it is going to be capital R, or small r, small r. In the case of heterozygous condition, one will be capital R, one will be small r. That is going to be your heterozygous condition. Clear, all of you? Then there is something called as dominant allele, one is recessive allele. Remember, students, there is something called as dominant allele. Dominant allele is also called as your wild type. It's called as wild type. Then there is something called as your recessive allele. There's something called as recessive allele. Now, recessive allele is called as your modified type also. It's also called as what? Modified type. Now, remember, students, the dominant allele will always express. Even in the presence of other allele. That is called as dominant allele. For example, in capital T, small t, capital T is going to be your dominant allele. Now, what about recessive allele? Recessive allele is an allele which cannot express itself at all in the presence of dominant allele. For example, in capital T, small t is going to be your recessive allele. Small t is going to be your recessive allele. Clear? And remember, if only when small t is there, in those conditions, recessive allele will be able to express. Otherwise, recessive allele cannot express itself at all. Okay? Cannot express at all. Now, can I ask all of you a question? Who is known as the father of genetics? Tell me that quickly. All of you, please smash that like button first, because no one teaches you such basics, and these basics are going to be very important for you to understand your next step. To be able to solve numericals, you need to know what is homozygous dominant, heterozygous dominant. See, this is called as your heterozygous dominant. This is homozygous recessive. It is homozygous recessive. Capital T, this is homozygous dominant. It is going to be your homozygous dominant, heterozygous dominant, and homozygous recessive in nature. The father of your genetics is none other than Gregor Mendel. Now, Gregor Mendel's main work was on hybridization, and he worked on a plant called as your pea plant. But if you look at your pea plant, there are total of your 7 contrasting characters. Look at here, 7 contrasting characters. 1, 2, 3, 4, 5, 6. One is missing. The missing one is going to be your height of a plant. It is going to be height of a plant. Now, will they ask you this table in examination? Yes, they will ask you. But how will they ask you? They'll ask you in a very simple fashion, telling that which of the following are said to be dominant in nature. Which of the following are dominant? And there is a small trick for it, that is going to be your gravity. That is going to be your gravity. That is green. That is going to be your pod color. Pod color, green is dominant. Green is dominant. Round is dominant here. That is going to be your seed shape. Round is dominant. Seed shape. Then we have A, that is axial arrangement of flower position. That is flower position, right? Flower position. Then we have V. V is nothing but your, where is it? Violet. Flower color. Flower color which is dominant. Flower color. Then we have I. I is nothing but inflated pod shape. See, inflated or full. Inflated or full. Inflated or full. That is going to be your pod shape. Pod shape. Then we have your T. T is nothing but your, where is it? Here. Not given here. That is going to be your plant height. Plant height is not given here, so I am writing it separately. Plant height. That is going to be tall. What about Y? Y is the last one. Y is the last one, that is going to be your seed color. right? Seed shape is there, then we have seed color. Y is going to be seed color. So all of these are your dominant. All of these are nothing but your dominant characters. Clear? Yes? All of you clear with this? They can ask you directly. They can ask you directly which are the dominant characters. Clear? The next thing I want all of you to understand is the monohybrid cross. I won't teach you the entire cross. I'll just teach you some few lines. Monohybrid cross. Because you have done this cross in your class 10th, you should be able to tell me what is the phenotypic ratio. What is the phenotypic ratio in the case of your monohybrid cross? Can anyone tell me? What is the phenotypic ratio? Phenotypic ratio is going to be 3:1. The 3 is going to be tall, 1 is going to be dwarf. What about genotypic ratio? Genotypic ratio? Genotypic ratio is going to be 1:2:1. 1 tall, 2 tall, and 1 dwarf. Right? 1 dwarf. Clear? Then you have something called as your incomplete dominance. Then you have something called as incomplete dominance. Incomplete dominance is a condition in which a particular dominant allele is not able to completely suppress the effect of recessive allele. So recessive allele is also expressing here and there. It's coming up. So it happens in an example called as Snapdragon or Mirabel's Jalapa. So there is incomplete dominance. So your dominant is not doing work. Basically, your dominant is not doing work, and because of that, your phenotypic ratio is changing. Phenotypic ratio is changing completely. Phenotypic ratio in the case of incomplete dominance is going to be 1:2:1. 1:2:1. 1 tall, 2 pink, and 1 red. Right? Now, what about genotypic ratio? Genotypic ratio is going to be again 1:2:1. 1:2:1. 1:2:1. This is going to be your homozygous dominant, heterozygous dominant, and homozygous recessive. Homozygous recessive is going to be same. Now, what about in the case of codominance? In the case of codominance, there are two dominant alleles. There are two dominant alleles, and both the alleles are able to express itself. Both the alleles are able to express itself, and there is nothing called as recessive allele here. Both are dominant alleles. And here we see a concept called as multiple allelism. Multiple allelism. Example is going to be your ABO blood grouping. ABO blood group. Your blood group. Blood group. Clear, all of you? Then the last one we have something called as your before we go dihybrid cross, do all of you know what is a test cross? Do all of you know what is a test cross and back cross? Do all of you know that? Should I teach that also? Do you know? If you know, let's not go waste on time on that. Let's go to directly in advance to that. Let's go to dihybrid cross. Go to dihybrid cross. In dihybrid cross, there are two genes involved. In the case of your dihybrid cross, there are two genes involved. Yes? The two genes involved, can anyone tell me what is the phenotypic ratio? What is the phenotypic ratio? Dihybrid cross. In the case of dihybrid cross, it's going to be 9:3:3:1. Now, what about the genotypic ratio? Now, someone commented in the last video, sir, can you explain the genotypic ratio? How to tell, how to identify? How to identify the genotypic ratio? Very simple. Very, very simple. So I'll tell you the small trick here which I've been teaching all my students. So first thing you need to do is take two genotypic ratio of monohybrid cross. So what is dihybrid? Dihybrid cross is typically nothing but a cross between two monohybrid. A cross between two monohybrid, right? So if you look at if you want to find the genotypic ratio of dihybrid, the simple trick is going to be, take the genotypic ratio of two monohybrid cross and just multiply it. What is 1 into 1? What is 1 into 1? 1. 1 into 2? 2. 1 second. 1 into 2 is 2. 1 into 1 is 1. 2 into 1 is 2. 2 into 2 is 4. 2 into 1 is 2. 1 into 1 is 1. 1 into 2 is 2. And 1 into 1 is 1. This right here is going to be your what? Genotypic ratio. Genotypic ratio of your dihybrid cross. Genotypic ratio of your dihybrid cross. Very easy, right? Very easy concepts. These are very easy concepts because in this chapter there are very few concepts which you need to think a lot. Few concepts you can easily find out. For example, the numericals which you get here. The numericals which you get here are very difficult to solve sometimes. But if you are in my student, you should be able to solve all the numericals, and I'll make sure of it again this year also. Okay. Now, apart from that, there is something called as chromosomal theory of inheritance. Chromosomal theory of inheritance. Chromosomal theory of inheritance. Chromosomal inheritance was given by whom? Can anyone remind me? Chromosomal theory of inheritance was given by whom? Chromosomal theory was given by your Sutton and Boveri. Sutton and Boveri in the year your 1902. In the 1902, Sutton and Boveri gave the chromosomal theory of inheritance. Chromosomal theory of inheritance is very simple. It is very, very simple. It tells you that the behavior of chromosome and behavior of your gene is same to same. That's all. Behavior of a chromosome and behavior of a gene is same to same. That's all. But the scientist who gave the proof to the chromosomal theory of inheritance was none other than your T.H. Morgan. T.H. Morgan gave us the scientific proof of it. Scientific proof of it. Okay. T.H. Morgan gave the scientific proof of it. The next concept which I want everyone to understand and where the numericals have come from, that is going to be your linkage. That is going to be your linkage. Very easy concept again. Very easy concept. I'll explain with the help of a small diagram and you will understand it very easily. Right? So imagine this is a chromosome. This is a chromosome. On this particular chromosome, we have certain genes. Now we have one gene is here, one gene is here, and one gene is very far. One gene is very far. Now let's call it gene A and let's call it gene B and let's call this gene C. Now tell me, is gene A and gene B are very close to each other? Yes, very close to each other. But if you compare gene A and gene C, they are very far from each other. Yes, very far from each other. Now, listen to me very carefully. Whenever they are since they are very close to each other, right? Since they are very close to each other, can I say linkage between your A and B is very high? Yes, sir. Linkage is going to be very high. Linkage is going to be very high. Now remember students, whenever on one chromosome, when the two genes are very close to each other, their linkage is very high, very close, link is very high. If the linkage is very high, can they twist and turn and do some recombination? No. So when the linkage is very high, your recombination becomes very low. Recombination becomes low. Similarly, in this case, can you see? In this case, linkage, are they close to each other? Gene A and gene C? No, they're very far to each other. So in this case, linkage is going to be very low, but recombination is going to be very high. Recombination is going to be very high. Clear? So by this, can we do a simple formula? Simple formula, that is, linkage is inversely proportionate to recombination. Inversely proportionate to recombination. Clear? Very easy. Your linkage is inversely proportionate to your recombination. Now there are a lot of numericals here and a lot of formulas. Actually, a lot of formulas. So let's not do all the formulas right now. We'll stop at this right now here. Okay, we'll stop it here. Clear, all of you? I believe there should be no doubt here. Now there are other concepts in this chapter, such as your pleiotropy, where one gene has multiple expressions. Pleiotropy, right? Pleiotropy is there. And there are so many concepts such as your chromosomal disorders. Yes, there are certain concepts like chromosomal disorders, such as here. Can you see? Your Down syndrome, Klinefelter syndrome, and then there is Turner syndrome. There are certain Mendelian disorders. Yes, Mendelian disorders, thalassemia, sickle cell anemia, as well as your color blindness and hemophilia. So students, you need to learn each disease one by one. So you need to make a complete table, one complete table where you divide chromosomal disorders and Mendelian disorders into autosomal as well as sex-linked. That table we'll do in the detailed class, right? Apart from this, there is one more concept called as pedigree analysis, which is very simple again, okay? So that marks the end of your genetics. Now apart from this, I'll be teaching you, hopefully, the entire your molecular basis, where the main questions have come from your replication, transcription and translation. These three topics are going to be very important for you to understand, and we will learn with the help of a lot of diagrams here, okay, as well as human genome project. And here we will learn a lot of scientists and their work. Search for genetic material, Griffith experiment, yes, Hershey and Chase experiment, Michelson and Stahl experiment. All those experiments comes under search of genetic material, right? Now, how do you learn microbes chapter? Sir, is there any trick to remember this? Students, remember, your microbes chapter is a complete chapter based on examples, and they will not ask anything apart from examples. And how do you remember examples? With the help of mind map. For example, if they ask you, they'll ask you microbes in household. So you have to look at the mind map and go here. Curd, lactobacillus. Bread, Saccharomyces cerevisiae. Then we Swiss cheese, Poprani shermani. Then we come to your again sewage treatment, and it's going to be separate category itself. Enzymes we have lipase. Organic acid we have acetobacillus niger, citric acid. Acetobacter we have aceti. Clostridium butyrium, lactobacillus, see, lactic acid. So this particular table or this particular flow chart will help you to map out every single microbe example, okay? So I'll be giving it to you, don't worry, the entire thing. Then last part of your class 12th is going to be your ecology. Now sir, what to study here, students? Remember, the first chapter, your organism and population, the main important part is going to be your types of population growth. For example, we have two groups. One is your logistic growth, one is exponential growth. Here you need to know the formula. This year NEET examination, before the NEET examination, every single year, this particular formula has been asked repeatedly. For example, in the case of your sigmoid curve, that is for your logistic growth, there is something called as your K. They asked you, what is K? K is your carrying capacity. Yes, simple question. Now, apart from that, they can ask you something called as population interaction. Now, in this population interaction, you need to know examples. More than this plus or minus or what is benefited, you need to know the examples. For example, in your predatory, in predation, they might ask you, a particular bird is eating an insect, Bird is eating insect. Is it predation? Yes, it is a predation. A herbivore is eating a plant. Is it predation? Yes, it is predation. So you should know examples of each one of them. Clear? Examples are going to be very important here. Now, when it comes to your ecosystem chapter is a chapter where questions will come from a single line. For example, this year examination you got question of productivity. Even though the question was little questionable, the question came from productivity. Similarly, questions have come from something called as your decomposition. They can ask you the properties of your humus. That is the important question, right? Then there is something called as ecological pyramids. We have in total of 3 types of pyramids: pyramid of number, pyramid of biomass, and pyramid of energy. But will they ask you directly the pyramid? No. What will they ask you? They will ask you exceptions. Exception in your pyramid of number is going to be a big tree. Pyramid of number can change there. That is, pyramid of number can become inverted. In the case of pyramid of biomass, the exception where the pyramid is inverted, that is going to be your ocean ecosystem, water ecosystem, where pyramid of biomass is inverted. What about pyramid of energy? Is it inverted? Any case? No. Pyramid of energy is never inverted. Pyramid of energy is always unidirectional. So remember those points. Remember those points. In the last chapter, that is your, in the last chapter, they will always ask you something called as species area relationship. Species area relationship. You need to know the entire formula. They'll ask you what is S, what is C, what is Z, what is A. Entire species area relationship should be known to you. What is species area relationship? That is nothing but the more you search, the more organism you will find. Only up to a certain limit. That's all. The last part is going to be your causes of biodiversity. There's something called as evil quadrat. There's something called as evil quadrat. Now, what is this evil quadrat? Evil quadrat are nothing but the main causes of biodiversity loss. For example, the first one is going to be your habitat loss and fragmentation. Habitat loss and fragmentation. Habitat loss and fragmentation. Then we have something called as your overexploitation. Here we learn about the soybean cultivation, and it is the most important one. We are in overexploitation, we learn about stellar sea cows as well as your passenger pigeons. Overexploitation. Then we have something called as your alien species invasion. Your Lantina camera, Parthenium, and water hyacinth. All of them are part of your alien species invasion examples. Students, examples are the most important one. The last one is going to be co-extinction. This is mostly seen in the case of your plants as well as certain pollinators. The pollinators is gone, even the flower is also gone. Co-extinction. The last part is going to be your the specific dates here. Oh, this is the one before that. Something called as in situ and ex situ conservation. Again, a controversial one. In situ and ex situ conservation. That is very important. Then we have your dates. First one is going to be your Rio de Janeiro in 1992. And the aim. Aim is what? Appropriate measure for conservation of biodiversity and sustainable development. Then we have the World Summit which happened here in Johannesburg in 2002 in South Africa. What is the goal of this? A significant reduction in the current rate of biodiversity loss at global, regional and local level. Students, there it is. That is going to be your entire class with biology in a gist. Entire class with biology. I know I didn't explain molecular basis, but that will come. And this is from your botany part. This is from your botany side, right? So students, I know there have been a lot of confusion going on regarding re-NEET or happening. If you have any confusion and you want to have a certain, importance, like, sir, can you tell me what is going to happen? And you need someone to talk to. And if you want to talk to Shreyas sir, there is a helpline number. We have made a helpline number. If you go to the description of this video, you will find helpline number. And you can always talk. That is free one-on-one mentorship and clear all your NEET related inquiries with Shreyas sir and the rescue team. Right? So that is going to be there in the description. So go click on it and you will get it. And there it is. Your entire class with biology in a small gist. So let me know in the comment section of today's video how the session was. If it was useful to you, comment it down. And also quickly smash that like button, all of you. Quickly smash that like button. And if you were able to understand something and you were able to clear your basics here and there. Okay. So with that being said, I'll take off right now. And take care all of you. Bye bye and have a nice day. Bye bye all of you.